Okay, let's have a class. So, you have Staphylococcus aureus, Streptococcus from your throat. Is that right? Yeah, some will find it. That's why I told you it's a very exciting material. Today we have a more exciting material right here, okay? But we're going to talk about this later on. Fact sheets. You already took all the fact sheets already, gone? Okay, I'll need to bring some more. Okay, I'll need to bring them more later on. Now let's talk about the results. Let's talk about the material. We will have 3 lab today, lab work. 2 biochemistry tests, and 1 is a pathogenic fungi. We will do the observation. Okay, so let's talk about biochemistry. So biochemistry test number 2. This will be number 2. So the first test is SIM test. Okay, this test is a little bit different from what we did before. Instead of using loop, we will using is a stab or stab loop, which means there is no circle there. No circle on the top. Okay? And we were not using a traditional medium. What we will use is a semi-solid medium. It's a little bit different. Number 1, stab inoculation. Number 2, semi-solid medium. So how we do this? You will get 0.5% semi-solid agar. And what you will do is, from you assign the culture using a stab loop to touch it, and then go down all the way to the bottom, right in the middle, and took it out. Very simple. So you go down in the middle, and then you took it out. This is we call 1 stab, 3 jobs, or 3 shoots. Okay, what are the 3 jobs? S indicate sulfide. I is indo. M indicate motility. So let's talk about those 3 things. Number 1 is motility. Okay, first thing, motility. M indicates motility. This is easy. Okay, how do we know bacteria is motility positive? After you doing the stab inoculation, if you see the bacteria goes everywhere, which means the whole tube has a bacteria growing marker, that is motility positive. If you see, after you stab inoculation, your bacteria only stay in the middle, like this, staying in the middle, this is, we call it negative, motility negative. Now, how do we know bacteria have a strong motility or not? We based on these results, we also based on, if you still remember, the location and the morphology of the flagella. So if you have only 1 at the end, we call it a monotracheous. If you have 1 at each end, what we call it? Is that called amphitrichous? Amphitrichous. If we have lots of stack on one end, we call it a lophotrichous. Is that right? Lophotrichous. How about if we have everything goes everywhere? Petrochemicals. No, petrochemicals, it should be O. So we know the motility, which is real movement. So that is the first test. M is easy. Second one I want to mention is S. S is sulfide. What this means? Remember, electron goes to oxygen. We call it aerobic respiration. If electron transfer from sulfide reduced to H2S, this is called anaerobic respiration. So anaerobic respiration. Some of the bacteria can use sulfide as an energy resource and the final electron acceptor. Now, how do we know the sulfide is positive? In this medium, there is some metallic ingredients. For example, it may have ions there. If sulfide reduced to H2S, it turns black. So it is very simple. If on next week, Tuesday, you see this tube, the whole thing turns black, it is sulfide positive. Okay? So that's the second one. Last one, Indo. What is Indo? It's a little bit difficult. I indicate Indo. So little organic chemistry. We need to know the structure of amino acid. What is that? Amino acid is always NH2, CH, COOH, carboxyl group, amino group H, and the all side chain is different. Okay, here the all side chain is a very special. What is that? It is CH2 goes on top is like this. Then you have NH. What is this amino acid? Can somebody tell me? Tryptophan, very good. I believe that's he's a very good student. Tryptophan. Okay, we'll talk in the lecture, and that will be the extra points right outside the chain. Okay, so this is tryptophan. Now, some of the bacteria can use amino acid as an energy resource, not like glucose. They can use amino acid. What they do? They will break down right here. Okay, then generate this big chunk of stuff. Now, accurately say, it should be this area. This area, okay? This is Indo. So what is Indo? Byproducts of tryptophan. Now, how do we know the bacteria generate Indo? We need to add a very special reagent called COVAC. A very special reagent called COVAC. And after add COVAC, what's going to happen? it turns cherry red. So this you have to do a supplement test is next week Tuesday. After it grows, we need to add a drop of Kovac reagent on the top. If this guy here turns cherry red, then it is Indo positive. Okay, so that's very simple. That's why this test is very easy. You just put in, took it out, incubate. Of course, we have to do incubate. 35 degrees Celsius, 24, 48 hours. We see the color change, but you need to know the story behind it. It's a lot. It's testing motility, testing sulfide, and testing indo. And some of them are biochemistry behind it. So that's why it is a little bit different. Okay, a little bit difficult. Next one. This is a very simple slant. Looks like it's the same. What is this test? So test number 2. Phenylalanine deaminase test. Okay, same thing. This is tryptophan. Do you know the structure of phenylalanine? NH2, chiral C, carboxy group, COH, and the side H. Then on the top, what we have? CH2, and then is a phenyl group. Like this. That's phenylalanine. Okay. PHE, just say phenylalanine. What is deaminase? Is this guy is took out? Is gone. So left side, what is that? Phenyl pyruvate acid or phenylpyruvate. Now, why this is important? Some of the bacteria could use phenylalanine because when they have a deaminase, took out of the amino group, the left side right here becomes phenylpyruvate acids. Do you remember pyruvate is an important chemical originally coming from glycolysis, and they go TCA cycle, or go fermentation, or go other things we didn't even know. Phenylpyruvate acid is a very similar function as pyruvate. They could also go modified glycolysis, modified TCA cycle or different type of the fermentation. They do whatever they like based on different situation. So that gives them intermediate biochemical stuff. They could be further survive. That's why it is important. Okay, how do you know it is phenylpyruvate acid positive? How do you know it's deaminase? So the work is very simple. You get a slants, and then you do a zigzag streak. How do you know it is positive? And we should do confirmation test, which is next week. We need to add a drop of FeCl2, ferric chloride. If this area turns dark green, then it's a phenylalanine deaminase test positive. So it is phenylalanine deaminase test positive. Okay? So today's first two work is very simple. Everybody gonna grab two testing tube. This little tiny one is for SIM test. Using stab inoculation. One stab, 3 jobs. The second is the slant for phenylalanine deaminase test. And what you should do, the first thing is label, don't forget. Label on the top is your name, bench number, and the bacteria, what you put there, okay? Now, we're gonna do the same, assign the bacteria. We're using the same thing, E. coli, Bacillus, Lactobacillus, Citrobacter. So every bench, the first two of you could do E. coli, followed by Bacillus, Lactobacillus, and the last two do Citrobacter. So E. coli, Bacillus, Lactobacillus, Citrobacter. Okay, the same thing here. E. coli, Lactobacillus, E. coli, Bacillus, Lactobacillus, and Citrobacter. So E. coli, Bacillus, Lactobacillus, and Citrobacter. Okay, not done yet. We have big stuff haven't talked yet. Very molded Asian meatball. It has been there for around like two months already in the back of my car. So they're very molded. So what are we gonna do? We will do, based on this fact sheets, we'll do the observation, what type of the molds it is. We haven't talked about the molds too much. Okay, so this is another one we want to talk about is pathogenic modes. And we need to know a little bit, okay. This is like, let's say, modes. Modes, when they generate, it is through broad base body. And this is very similar to like your ovoid. Ovoid, okay? And usually this is large. This is about 0.5, I would say 0.5 micrometer sometimes, even more than that. Sometimes we have 500 micrometer. So it's larger than the bacteria. And we also call it gram stain blue, not say positive or negative. And then most of the time the structure, what it looks like, very heavy, fuzzy at the bottom. We call it some mycelium. And then you're gonna have these things comes out. And these type of thing, like a flower, like a seeds, they're gonna be released. This is called conidia. Conidia. They will be released. And in this area, in the Ohio River area, in Kentucky, West Virginia, Ohio, what happened is there is some of the pathogenic fungi carried on by the birds dropping called the histoplasmomycosis. If you inhale, you're going to have some of the coughing, sneezing, some type of like allergy, but not really. It will be cause lung disease. So just want to let you know that. Now we had a very molded this bomb. What are we going to do? We are using a very simple test, not difficult. So we got a glass slides. Right on the glass slides middle, we're going to add one very special dye, is lactophenol cotton blue. We just put out one big dye. With the dye is blue color. And then we're going to be covering this using a tape. And this tape will touch a very molded bond. Let's just say bond. or bread, we say. And then you cover it. Underneath microscope, we're using 40x transfer to 100x. And most of the time you use 40x to see the morphology. Now, why the morphology is important? And look at your fact sheets. On the first page, we are trying to find whether you could see those 3 typical pathogenic modes, which is number 1, right path, number 2, aspergillus, and number 3, penicillin. Those are the 3 we want to find. And you can compare the morphology, which is on here, and determine what type of the modes it is. Now, how to do this? You absolutely need to wear gloves. Then you come here. And we need using a tape. The tape will be like this. And you touch it and have your glass slides ready, then you press it. In the middle of the glass slides, we'll have a lactophenol cotton blue. And then you go ahead to do the observation. This one you could do is 2 of you have 1 representative to do, because it is really molded. So only 1 of you from each group come here to get it, and you 2 of you can share and take a picture or draw on your notebook. You will get a brief idea what type of the pathogenic modes on this very molded bark. Okay, so those are 3 works gonna do. So let's do that. Everybody come into the front. Let's do this pathogenic modes first. Then you do the biochemistry test.